currently the bane of our clinical existence, clinical standpoint, as well as the microbiologist standpoint, is drug resistance. It's a bit of a sore point. Why drug resistance has become such a problem. There are certain people out there, some microbiologists, some non-scientists, who say the overuse of antibiotics in the farming industry is the reason why we have drug resistance everywhere. There is some validity to that, but even with the validity, the source of the reason for pointing it all towards the farming industry is overblown. For those of you who've never been on a farm, cattle farm, sheep farm, chicken farm, whatever, since they have so many animals in close proximity to each other, if one gets sick, chances are whatever is causing to get sick, viral, bacterial, can then be quickly passed through the rest of the herd, the flock, whatever. So with the development of antibiotics back in the 50s, 60s, farmers would just prophylactically give, the cows, the chickens, the goats, whatever, here's just a bunch of antibiotics to keep you from getting sick. Overuse, yes. Incorrect use, yes. So what that did is it selected for strains of the bacteria that were resistant. Because remember, wipes out 99.9% of microbes present, blah, 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 that stuff that we've mentioned before in this lectures and other lectures. You wipe out 99.9% of a million, that still leaves you 100. Okay? So you take that 100, you wait 24 hours, and you're back to 100 million. Here's the next treatment. Wipe out 99%, you're back to 100. Wait a day, you're back to 100 million. You just keep going back and forth like that. What that does, though, is it strengthens the resistant. Day after day, treatment after treatment after treatment of not wiping out all but only some, means that the microbes you started with, the bacteria you started with, even if it's the same species, but 3 weeks later, here's what you have now after daily inaccurate usage of the antibacterial, means that you have strengthened the resistant. What you have 3 weeks later is no longer the same strain. You now have a strain that, that antibiotic probably won't affect at all. You have selected for resistant strains. Those resistant strains spread to humans, blah, blah, blah. Even if those bacteria can't grow on us, in us, because remember the Goldilocks rule we talked about back in the bacterial chapter, too hot, too cold, just right, too high, too low, just right pH, all those different things. But as long as they're there for a little while, thanks to conjugation, the resistant bacteria from a cow that's on you, in you, it's only going to be there for a day before it gets wiped out by the immune system, is going to conjugate with one of your normal flora bacteria, passing over any genes that aid in resistance. That's how they think it started. Is there some validity to this idea? Yes. Is this the only reason why we have such a resistance problem? Oh God, no. It's, it's a factor, but it is not the only reason. Go back to the last lecture, and I'd said, mommy, daddy, bring in little Johnny, little Susie's got the sniffles, blah, blah, blah. Mom and dad can't afford to take another day off work, so give little Johnny, little Susie an antibiotic that'll clear this up now. Misuse, the 50s, the 60s, the 70s, the 80s of drugs. Well, oh, you got the sniffles. Here's an antibiotic prescription. Oh, you got a swollen whatever. Antibiotic prescription. Misuse of the antibiotics by us. Also selected for more resistant strains. Where does this resistance come from? Well, like I said, it's, starts off as a spontaneous mutation. The target, the drug target, the gene that produces the protein that's, the metabolic target of the drug. Mutation slightly changes the shape. The drug can't interact as well. Its efficacy has now been lowered to 75%. It's only 75% effective, 50% effective. One bacterial mutation in one bacterial cell out of 100 million, a billion, not that big a deal. Statistically, it's insignificant. Until you bring in a drug that's going to wipe out anything that's not resistant. So instead of 100 million, a billion, you're now down to 1,000. And of those 1,000, one was resistant. One bacteria becomes 2, 2 becomes 4, 8, and so on. A day later, you go to take the drug again. That one resistant bacterial cell now has 1 million that are just like it. Take the drug again. And again, and again, and again. You wipe out everything that's susceptible, and all that's left are bacteria that are resistant. And then the second thing it shows there in red, acquisition of new drugs, or new genes, sorry. That's that conjugation. You're going to pass over a plasmid, and on that plasmid is a gene, penicillinase, we talked about previously. A gene that's going to allow for a greater resistance. So you look at drug-resistant mechanisms as it's shown here in table 10.9. Either a direct enzymatic attack, the penicillinase, or it's going to be the ability to stop uptake of the drug, or you now have a pump that allows the bacterial cell to pump the drug out before it can have any effect. Then it's just flat out change of the target. You look at the example there showing the ribosome. Erythromycin, clindamycin specifically target the ribosome. They interfere with protein synthesis. Translation cannot occur because the ribosome either can't take up a new tRNA or it can't translocate from one codon to the next. Well, drug resistance is going to be a slight mutation in the ribosomal RNA gene, which is going to get a slight change in the shape of the ribosome. Nothing dramatic, but just whatever portion that erythromycin or clindamycin we're going to attach to, no longer there. Drug has no effect. Got to remember, drug resistance is a natural thing. These bacteria, these microbes, have been here much, much longer than we have. They've been in constant battle not only with each other, but with fungi for space, for nutrients, for water. They've developed their own weapons, which we now think of as antimicrobials. We use as antibiotics. And they have developed their own defenses, penicillinase, things like that. Figure here shows you what I was talking about when it comes to natural resistance selection. All the microbes, the normal microbes, the bacteria, are in white. One of them, two of them mutated. They're red. They now have slightly more resistant capability to the drug. Drug comes in, as you see in number 2, wipes out all the susceptible. Pretty much all that's left are the ones that have a slight resistance. They're the ones that get to grow. They're the ones that get to repopulate. You go take the drugs again, you wipe out the ones that were only slightly resistant or completely susceptible that are still there. The ones that have a greater resistance are all that's left. And you keep selecting and selecting and selecting for greater and greater resistance till all that's there now in your throat are drug-resistant Streptococcus pyogenes. What's even worse, as window 4 is showing, horizontal transfer, conjugation, allows for the passing of a plasmid or a chunk of genomic DNA from a resistant to a non-resistant. That's also a problem.